Hi everyone, this is Barnabas from Alligator Bioscience and welcome to my poster where I will take you through RUBY, a novel bispecific antibody format that allows straightforward generation of new bispecific antibodies from any two monoclonal antibodies. Briefly, RUBY bispecific antibodies are of the appended IgG class of bispecific antibody formats. Fab domains are linked through their light chains to the C-terminal end of their IgG molecules, and this presents several benefits. The lack of the single-chain fragment components allows for a plug-and-play capacity, meaning there is no need for lead optimization, and new bispecific antibodies can be generated in shorter timelines. The fusion of the appended Fabs through their light chain solves mispairing problems where products of correct sizes but with mispaired light chains cannot be produced, and this makes manufacturing and downstream processing of RUBY bispecific antibodies a straightforward process compared to many other formats. Presence of the Fc domain allows for tailored Fc interactions and the possibility to fine-tune interactions with Fc neonatal and Fc gamma receptors for optimal half-life and functionality. The tetravalent architecture allows for retained bivalent interactions against each target and more natural antibody-antigen binding kinetics. We have produced some bispecific antibodies using this format and several evaluations have been made. To start with, binding was assessed using ELISA and Octet, where we show good binding of RUBY bispecific antibodies, similar to that of the monoclonal antibodies. We have also been able to show that RUBY bispecific antibodies are able to activate T cells and B cells only in the presence of tumor-associated antigens. To the figure on the left, a functional CD8 T cell assay was performed using primary human CD8 T cells cultured with titrated concentrations of a RUBY bispecific antibody in the presence or absence of the tumor-associated antigen coated onto the surface of microtiter plates. Interferon gamma production was determined by ELISA as seen in the blue and green curves. The results are from one representative individual donor. To the figure on the right, primary human B cells were cultured with titrated antibodies in the presence or absence of a tumor-associated antigen expressed on Schwann cells. After 2 days, expression of CD86 on B cells was analyzed by FACS as seen in the orange and green curves. The graph shows pooled results from 3 donors in one representative experiment of 2. We have also evaluated tumor localization and anti-tumor efficacy of RUBY bispecific antibodies, where we are able to show that these antibodies can efficiently localize to the tumor area where they bind the tumor-associated antigen expressing tumor cells, as well as another target expressed on immune cells, and are able to induce a strong anti-tumor effect. In these experiments, transient mice were inoculated subcutaneously with MB49 tumors expressing the human tumor-associated antigen or the same cell line lacking the tumor-associated antigen expression. In figure A on the left, we show how RUBY bispecific antibodies are able to localize in the tumor. Here, mice received a single target RUBY bispecific antibody in green, monoclonal antibody in orange, or the vehicle in gray, and analysis of the tumors for IgG positive cells was performed using flow cytometry after 24 hours. In figure B on the right, we show a strong anti-tumor efficacy induced by RUBY bispecific antibodies. Here, the mice received RUBY bispecific antibody or vehicle control treatment on days 10, 13, and 16, and the tumors were measured continuously until the first mouse in each group reached a tumor volume of 2000 cubic millimeters. In terms of pharmacokinetics, here we show one of our RUBY bispecific antibodies that displays a pharmacokinetics profile similar to that of a fully human IgG1 LALA mutated monoclonal antibody. We have also assessed manufacturability of these antibodies in transient Hague and Schwann cells, where good yields in the same range of a monoclonal antibody were obtained. We have generated stable Schwann cell lines where we have produced these molecules in titers above 1 gram per liter, and very low impurities after protein A purifications were obtained. These molecules can also show good serum stability and thermostability and can withstand elevated temperatures. Furthermore, these molecules show minimal degradation when subjected to various conditions such as freeze thawing, incubation at low pH, and shear stress. With that, I would like to thank my colleagues for their tremendous efforts towards this work and thank you for listening. Please don't hesitate to contact me for any further discussions.